Today's topic, because I'm a physician, is to talk about Nobel Prizes in Physiology of Medicine. I thought I would end up talking about multiple, but I found out that by doing my research that the first one is such a good story. I'm going to do the first one. And I thought I would start out just talking about Alfred Nobel. And we have to have a little science, sorry. We have to have a little high school science with Alfred Nobel, who made his loop the invention of dynamite. Mm-hmm. So first, let's this is Alfred. No, this is Alfred E. Newman. Mm-hmm. Okay, so this is Alfred Nobel, and he invented dynamite, which is based, of course, on nitroglycerin, which was synthesized by Ascanio Sobrero. Trip slightly off the tongue. And he wrote, "When I think of all the victims killed during nitroglycerin explosions and the terrible havoc that has been wreaked, which in all probability will continue to occur in the future, I am almost ashamed to admit to be its discoverer." So there you go. Because obviously it's very unstable and it explodes. And was
he referring to victims in his lab, or?
Yeah, and anybody who handled it.
Yeah.
And so here's this little science here. Here's the structure of nitroglycerin, and it begins with the old glycerol molecule, including nitric acid and sulfuric acid. And he gets these. This pointer comes through to us through courtesy of who children on his. This is free. This is from Microsoft. It's just Microsoft analysis. It's free at the Microsoft store. It's not good for doing work like on Word things. It's not precise enough for that. But just for gently pointing things out, the nitrates, it's a very good cursor. So it's free, so it's good to download and use it and turn it right off. So this is glycerol trinitrate. So it's NO2. It's really NO3 because you have the ester bond. So it's NO3. That's what makes it nitrate for your organic chemistry. And the key to it is the high energy state of these nitrogens. The way it works chemically is nitrogen has 14 nucleons, 7 protons, 7 electrons. And so remember from high school, the shells look nothing like this, the probabilities. The first shell has 2 electrons, and that's in a neutral state. And here's where the excitement comes with nitrogen, is the second shell. And it has, in its lowest energy state, the second shell has 8 electrons. But here it has only 5. This is the balance of the nucleons. Neutral charge. So it's 5 valence electrons. And what it does, it can either go, it's really AC/DC, it can go lose the entire shell, and it's a very high energy state, but the shell is empty. Or it can go completely the other way and fill the shell and be in a high energy state. Because now the one it's +5, here on the top one, it's +5, from the bottom one, it's -3. So this is very high energy states. These are for all dry explosives, but these are rockets where you don't use oxygen, you don't burn anything. It's dry fuel for rockets. They all use the same principle. And so here's where it wants to get to. It's the lowest energy state, which is N2, which is practically inert. 80% of the atmosphere is nitrogen. It just sits there. So I thought I'd show you how explosion works, but I didn't want to use a complicated thing. So I thought I would use Timothy McVeigh's model, but was it used in Dallas? Exploded that thing.
Oklahoma City.
Oklahoma City. Sorry, yeah. Oklahoma City. So he used ammonium nitrate. And if you look at the formula here, ammonium is 4 hydrogens, so it's a positive state. So 3 of those hydrogens have yielded their electron up to the ammonia. So the ammonia has 8 in its outer shell. And then the salt, the other side of the salt, this nitrogen has given up all its electrons. So it's in a +8. So that's really 2 huge different energy states. So when the reaction happens, you get the ammonia, I just balanced it, and the nitrate. goes to three nitrogen.
Okay.
Who's that? And three nitrates in a very low energy state, and then nitrogen dioxide in water. What happens is you get that. And if you look at it again, the ammonia is in a low oxidation state, and the NO3 is in a high state. So they come together to give them tremendous amount of energy. So what Nobel did with this unstable product is he stabilized it. And the first thing he did was he took 3 parts nitroglycerin, 1 part diatomaceous earth, which is really absorbent. Do you ever use that thing?
No.
Yeah, because it's very absorbent. Just keeps
We
everything.
knew about it, but never did.
Very keeps everything. It's just a white powder. It's cocaine. It's very stable.
Jerry, do we know how many, when he's experimenting to get the dynamite to make it kind of more stable in nitroglycerin, how many times he had to go through substances to find something that did what he wanted it to do? Does he disclose that at all, or did he just get lucky with that?
I don't know how lucky he got.
Okay.
But he was an inventor. He did lots of things prior to this. He invented all sorts of stuff. He was a ne'er-do-well, actually. He was going bankrupt all the time. He invent this, he invent that, he'd go bankrupt. He'd go contract and go bankrupt.
And made money on that.
And finally, he hit the jackpot. And the first thing he made was, he called it gelatinite gel.
Yeah.
He did. Got it. And this was nitroglycerin, nitrocellulose, and then wood pulp.
Staples.
Okay, yeah.
And it was very powerful and not brittle. So gelignite was the primary explosive. And he made also blasting gelatin, which was probably 90% nitroglycerin, gelatin, nitrocellulose. These flew around the stabilization. And all of his formulations collectively are just called dynamite. And he also invented the blasting cap. This is sort of key. In 1865, he took a copper capsule and filled it with mercury fulminate, which is very unstable, and high energy state as well. And let me show you how this breaks down. This is a blow up here of what's inside there. Here you have the fuse, and then you have the mercury fulminate here. And when the fuse hits burns the mercury fulminate, it goes bang, like that. And it gives a jolt. And then the jolt explodes the stabilized nitroglycerin. So here's the jolt here, and out here is the stabilized nitroglycerin, and that jolt is enough. Thing goes on. And that enables obviously controlled demolition, mining, tunneling, and structure, which is done, just drills and manually and facilitated everything. However, we want to talk about his son, Emil Nobel, and you'll notice the length of life is 21 years. He worked in the Nobel factory in Marienborg, Sweden. He was killed in an explosion. Nitroglycerin. He lost his son. Dangerous work.
Mm.
And then he set up the Nobel Prize in 1895. And I took this from their website. It's this symbol of excellence. And by encouraging innovation, he put up a monetary award. Here it is. In 1901, it was 8 million Swedish krona, which is about, just for inflation, it's $850,000. It's now $1 million, so it hasn't changed all that much. See the Nobel coin. And so the first one was in 1901. And it just happened to be given for the conquering diphtheria, which was scourge at the time. And remarkably reduced the disease and was the beginning of the understanding of immunology, the scientific work. Immunology was either not understood or not even recognized that we could be immune to things. Despite Edward Jenner's work a couple of centuries before, it just wasn't recognized that we could be immune. It's hard to understand that now.
In the first,
Jerry, the first Nobel Prize was in physics, and it went to Röntgen for X-rays, which predates your physiology stuff, and yet it's the foundation of modern medicine. So, yeah, I think that was an omission.
What was physiology medicine? That was a physical. So it was the beginning of understanding the immune response. But what we're going to do, so don't look it up. What we're going to do is we're going to take a vote at some point in the lecture after we've gone through the various people who contributed. We're going to take a vote who we think should win the Nobel Prize. They're not all that clear. They're not all that clear. And this is so I made this graphic for Charlie Holbrow. I think I used Inkscape. This is our graphic for the Lexington computer technology. You know, my father used to say that I have all the skills to put me in a low income bracket. So, so I can do this.
Very encouraging.
Well, I was interested in science, and he was very smart. He went to Harvard Law School, very smart, knew nothing about science. And when I would talk about things and explain them in science, he would look at me and say, "You are a compendium of useless information." Now, this is an old slide from a lecture I gave prior to COVID, a talk I gave prior to COVID. I just thought it would be interesting to see how smart I was at the time. Oh, you have, and this is the virtuous cycle of discovery. I just made this graphic. Where you have a discovery, and you have experiments and measurements, data, and people make a model, and they might make an application. But at the same time, people look at the data, they create more hypotheses, new equipment, new measurements, new data, and new models. And it just keeps getting better and better and better. And that's what scientific advancement is. And this is the pathway of scientific advancement, going around this virtuous cycle of science. So I should give a little background on diphtheria. But first, I want to just get the nomenclature, although I'm jumping ahead by 4 years, I'll show you why. Hans Christian Graham invented the Graham stain. Now, he didn't make anything new. He took existing products. Maybe he did Graham stains in high school. And what he used was, medicine. It's the line of medicine. He used crystal violet. And crystal violet stains all bacteria. And then if you wash it with ethanol, some bacteria yield up the crystal violet, become clear, you can't see them. And then you use saffron, which is a red-orange counterstain, which you can't see on the blue ones. You can't see it because it's overcome by the indigo of crystal violet. So this is a gram negative, meaning that a gram positive means it hangs on to the crystal violet, it won't yield it up with ethanol. And gram negative means it yields up with ethanol. And diphtheria is a gram positive organism. But this was in 1884. We're starting in 1880. This wasn't around then, but it's our language. So
Barry, before you go on, could you tell us what the symptoms of diphtheria are? What it would feel like if you had it?
Well, if you looked at the slide chart, you might actually know. Manifestations begins with upper respiratory symptoms, cough and sore throat. Progresses to necrosis, that means death, of the pharyngeal and tracheal membranes with the formation, this is an important point, thick, leathery, the word is leathery. And it may become invasive and affect the heart, liver, and kidneys. Those are the symptoms. That's what happens when you get diphtheria. And death from diphtheria is divided into two groups. In the adults, they do get necrosis of the pharynx. It's not obstructed, but it's invasive. They primarily get cardiomyopathy and cardiac sepsis and die of cardiac failure. That's the primary modus of death in the adult. In the child, under 5, where the airway is very small, they asphyxiate with the airway occlusion by this necrotic leathery tissue. And diphtheria is transmitted man to man. So you can imagine a family of losing 3 children, all watching your children asphyxiate.
Right.
That's what it's like. It was a horrid disease and greatly feared. So the death rate in the 1880s was unknown. They just didn't have the public health infrastructure to know the death rate. But it was a very common disease, and death was a common outcome. In the 1920s, when we started to have public health data in the United States, there were 12 to 15,000 deaths annually. The death rate was 7.5% among adults, which is much higher than COVID, much higher than COVID. And in children, it was 50%. So that was a gruesome disease.
These are figures in the United States?
In the United States, yeah. Now, diphtheria was first described way back by Hippocrates, like everything else, in 400 BC. There were various epidemics which were described over the years. In 1576, the diphtheritic membrane was described by a Frenchman, Guillaume Daillou. And in Spain in 1613, they had a huge epidemic. It was El Año de los Garrotillos, which is the year of the strangulation. Many people, children died in Spain that year. And then 1821 is really where it began. So I want to use modern medicine, observational medicine, where it was described in detail and was named diphtherie, which means leathery. And he performed the first tracheostomy for leaflet airway obstruction, which of diphtheria. Now here's the timeline I want to show you, and this will relate to voting. I'm going to show the same timeline again when we vote, so you don't have to memorize the timeline. So in the very beginning, to see bacteria, as I talked about with my co-talk, was Lowenhoek, who they say he invented the first microscope. It was really a magnifying glass, but he invented lens grinding. But this was really a discovery waiting to happen. Lots of other people in Europe were doing the same thing, right? And learning to grind lenses. He got famous, but well, that's not. And then the giant of bacteriology is Louis Pasteur. Louis Pasteur clearly would have won the first Nobel Prize in physiology medicine, but the rule was you had to be alive in 1890 when nominations opened up. He died in 1896, so this is 1901 was the prize. So it has to be 1990 nominations opened up, so he was ineligible. Because you probably don't want to give your first award to a horse. You probably want to give it to somebody alive and have a nice ceremony. Just my thinking. So he's not a candidate. Then we have Hanukaro, who you all know. He invented, which we'll talk about, methylene blue. That was the first stain used so you could see bacteria in the microscope. And we'll talk about it, because that was the beginning. So if you can't see it, you can't get started. So it's one of these pivotal inventions. And then we have Robert Koch, of course, who worked first with anthrax, with attenuation, immunization, strains, and his laboratory, show you, did a lot of the culture techniques and technology, which everybody is using even today. And then Edwin Klebs was the first, I'll show you when he did his first observation of the tuberculosis, of the diphtheria bacteria. And he worked with Friedrich Loeffler. Now, Loeffler was across town in Berlin. Loeffler was in Koch's lab. Klebs was across town, but they published together to show you. And then we have Wu and Yersin, who isolated the toxin. And then Barry and Kitasato in 1890, who made the antiserum. And that's where the voting ends, because 1900. But we're going to finish the story with the toxoid, because that finishes the story with Ramon in France, and then simultaneously Glennie. But let's start with the people who are candidates, since we're beyond Pasteur. We'll open them up. Edwin Klebs. But Edwin Klebs, now this can't be a picture of what he saw because he didn't have color photography. But this is diphtheria stained with methylene blue. That's what he saw. So what he did was he scraped the pharynx of people with diphtheria and put it on a slide and stained it with methylene blue. The key to curing any disease is you have to know the cause first. It doesn't come out of the sky. You have to know the cause of diseases until you get it cured. And this is really the first identification of the organism. But pontificating. Seeing the bacteria there, it doesn't prove that it causes disease. There's lots of diphtheroids. There's lots of diphtheroids in the pharynx. And they're just innocent bystanders, so they're there all the time. So the fact that you saw it doesn't mean a lot. It gets you started. It gets you going through that keyhole. So then we come to Frederick Loeffler. And he worked temporary, but let's work together. And the simple story that you get in biology classes, they discovered that the bacteria. Loeffler showed that the bacteria satisfied Koch's postulates in my last talk about what Koch did with anthrax, and then he applied the same thing to tuberculosis. He said, first you have to culture the organisms on culture plate. I'm sorry, first you have to get the organism from an animal that's died of the disease. It's got to be there. Then you have to be able to culture it on a culture plate. Then you have to take that, give it to another animal, and the animal has to die of the disease. Then you have to culture that from the animal on a culture plate and pass it along to show that it was the cause of the disease, that it's not an innocent bystander that you're just looking at. And they modestly named the bacteria Klebs Bacillus. Now let's look at the staining of it. By Heinrich Caro. Now I bring up Heinrich Caro. Heinrich Caro was a Sephardic Jew. And the reason I bring that up is that the people in the Pasteur lab were primarily Roman Catholics. And in the Robert Koch's lab in Koch Institute in Berlin, they were Lutheran. But at least we got one Jewish guy. So he's the Jewish guy. And remember from my last talk, well, he was a calico, he was a chemist, and he was calico staining in the dye industry. But Henry Perkin, I mentioned him in my last talk. Henry Perkin was an 18-year-old, he was 18-year-old college student. In a spring project, he was trying to synthesize quinine. His project failed. His glassware was stained with this indigo color. Stained everywhere, got in his hands, he couldn't get it off, got in his shirt, ruined his shirt, got caught, ruined everything. He immediately recognized he had a hot product. He had stain. Prior to that, indigo was the only real stain that was imported from India. It's a huge industry. He could make it from nothing in the lab. And he immediately went into the indigo business and making indigo, which was ultimately found to be the same aniline, which is a benzene ring with an amine on it. It was the same aniline that was in indigo. And it could be extracted from coal tar or potash, which is the boiled product of it's aniline, it has this indigo color. And then what he did, I should say that I looked up the religious background of people at the Koch Institute and the Pasteur Institute, and not a one of them had interest in religion. They were Lutherans, they were Catholics. I thought that was a sign of the times because religion was so important at that time. Who would go into science, which was sort of still a little anti-religious? People who didn't care. It was probably a small group at the time, but these people, every one of them had almost no religious affiliation other than just in name only. Very interesting at that time. So what Caro did was he took a benzene ring. The benzene is this ring here. If you take a hydrogen off it, it becomes a phenol. Changes its name. And he took thiazine. He made thiazine. He's a chemist. The thiazine has 4 carbons. It has a sulfur here. That's the thiol. And it has the azote here, the nitrogen. The original name for nitrogen was azote. And zotos means life. And when Priestley discovered oxygen, which sustained life, the other gas that was in the container was primarily nitrogen. The atmosphere is 21% oxygen, remained 79% some nitrogen. The trace gases weren't known at that time to be in the atmosphere. They had them, but they were kind of another atmosphere. And so Priestley found that oxygen sustained life, and everybody found out that nitrogen, the remaining gas, did not. Zotos is life, so it was azote without life. But then it was found out when they looked at the other gases, hydrogen and other gases, carbon dioxide, they didn't support life either. So azote didn't make any sense. I think there's no who changed the name, because nitrogen is used in the production of niter, which is for gunpowder. So it generates niter. So it's nitrogen. So it's nitrogen. That's how it got its name. So now we have the nitrogen there and the sulfur, and that's the phenol and the phenol. And he bound them together, and he made the phenol, is phenol, thiazine. And that's what phenothiazine is. So here's the main chromophore here, but he kept these ammonias on there to make it look like aniline. So it has a blue indigo color. It's mostly blue, actually. The chromophore makes blue. And this is what methyl blue looks Of interest, Caro thought that it had methylene. You remember from organic that methylene is carbon, 2 hydrogens, and 2 other bonds. But there's no methylene in methylene blue. So that's been around for 140, 150 years. It's never been corrected. There's no methylene in methylene blue. So there you go. You see why my father said I'm a compendium of useless information? Now you know what?
Do you know what the chemical reactions that he used to get methylene blue from those?
No, I don't.
But I'm wondering if methylene was used in that. Carry out those reactions.
I can look that up. In fact, I will look that up because I was very curious how a nice Jewish boy could make such a mistake. So I will look that up. That's a very interesting question. Very interesting. But very importantly, the phenothiazine ultimately made its way into the medical world many years later in the phenothiazine medications. It was used in the first generation antipsychotics, which were what we used. These came out in the 50s. And you may have heard of Thorazine. These aren't used anymore, first generation antipsychotics. They make the people feel like zombies. They say that's what they actually say. Makes them feel like a zombie. Thorazine, see the phenothiazine here, Stelazine, Mellarol, and many others were based on that. And the result was a huge impact. It permitted for the first time the control of agitated psychosis. And this is a graphic from a lithograph from Bedlam Hospital in England, where the people with agitated psychosis were just literally incarcerated with the change of laws. So just removal were done. And then in the 30s or 40s, I don't know what it was, they started removing the prefrontal lobe for people. They just basically put a scalpel through it. And so a lot of the craziness went away. With that, but they weren't wild. The prefrontal lobe was that. What's the expression? I'd rather have a bottle in front of me than a frontal lobotomy. Something like that. It emptied these hospitals out. It had an enormous impact. But I digress. So now, what's the problem here? He works in Bobby Koch's lab in Berlin. Robert Koch really invented the current culture techniques. What Pasteur, his competitor, the bitter competitors, had was a liquidy gelatin. So you could make a culture, but if all the bacteria fell in the gelatin, it's very hard to pluck out individual cultures for cereal, individual organisms, cultures for cereal cultures, because it's just liquid. And so Walter Hesse worked in his lab, his wife Fanny, mentioned this in another talk, brought up the substance agar, which is used in Malaysia to set up jams and jellies. And she suggested to her husband, that they use agar to create a product that would be firm and set up so they could culture the bacteria on top of it. Then you can pluck out a culture very easily into a subculture and a subculture and a subculture, a single colony. Because they had a lot of trouble with organisms overgrowing the whole thing. They didn't have bacterial suppression or anything, overgrow. And also in the Koch's lab was Julius Petri. And guess what he invented? The Petri dish. Now those things are used today. This is 1880, and they're used today. And Julius Petri died in his 30s, I think it was, probably of tuberculosis. Now Frieda left us in the lab. She said, "What's the problem with culture theory? It just goes over to Hesse and Julius and gets us agar, and you're all set." Right? Wrong. Diphtheria is a fastidious organism. It needs special nutrients. And ordinary cultures were overgrown by other bacteria and it didn't grow. It just wouldn't grow. So now what are you going to do? This is sort of the first little peek into immunology. And they don't know anything that you know now. And they say, what do you do? And so here's what Leffler did. He noted, and this is a happening observation, that Klebsiella bacterium, this bacterium, grew well in horse blood serum, while other throat bacteria was suppressed. Well, we know now that this horse serum, which is basically blood without the red cells, the serum contains antibodies. And what's the horse going to have antibodies to? The common organisms that we all share with horses and pigs and everything else. But it wouldn't have been exposed to diphtheria, so it wouldn't have antibodies against it. He didn't know this. This was just an observation, and a heck of an observation. And so that he could get the bacteria to grow, it was horse serum and some agar. And he put in meat broth for protein and egg yolk, which coagulates, it gives a little firmness to it and provides lipids. And then he added a little dextrose for energy. Well, that's a heck of a thing to figure out. Now he's got the organism and he can grow. You can't find a cure of disease until you find the cause. And now he can not just see it like Edward Plebs did, but he can grow it. And they published the paper together. But he gave the bacterium to guinea pigs over and over and over, and of course they all got diphtheria. So it satisfied Koch's postulates. So he knew he had the cause. So now he's thinking of the cause. Now we can work to a cure. Now this is the sick transit gloria department, renamed the Klebsiella bacteria. Seems that Klebs and Leffler ran into Karl Bernhard Lehmann and Otto Rudolf Neumann. And they were bacteriologists charged with naming bacteria for taxonomy. And they changed the name of Klebsiella bacterium to Cronibacterium diphtheria. They changed the name without permission. So what's in the name here? This is Cronibacterium diphtheria. That's the Corine is Greek for a club or a mace. To me, these look like dumbbells, but here's the club here, I guess. So they call it a mace. So it's corine. And diphthera is leather. So corine is a leathery mace. So Corynebacterium is the mace bacterium of leather, related to leather. So it's taxonomic rather than honorific. So that's a kick in the teeth if you're identified one of the greatest scourges of mankind. Your names have been enshrined forever, and what do you get? Well, Klebs gets a second tier gut flora, Klebsiella, and Fritz Lepser gets a rare immunologic pneumonia called Runzie is symphylis, the allergic cells causing symphylis pneumonia called Leffler syndrome. So there you go. And that's a kick in the teeth if there ever was one. So let's talk about diphtheria toxin, which is well characterized now. The pathogenic diphtheria release a toxin which prevents protein production by blocking ribosomal function. So cells are making enzymes all the time. Can't make enzymes, cell dies. That's how it does you in. But not all diphtheria organisms produce the toxin. And the tox gene must be present. And it's introduced and passed along by a phage. A phage is a virus. The bacteriophages are attack viruses. They're very specific. And so the bacteriophage for diphtheria carries the tox gene and can insert it into cells that it doesn't kill the cell. The tox gene is in the cell. Now, what's interesting is that it could have been a problem, is that bacteria have a very small genome because they're very small. And they can't have a lot of baggage like we can with our genome, quiescent genes. They just can't carry them around. They don't have room for them. And they're forever tossing out genes, getting rid of them, seeing if they can still survive. So it's sort of an efficiency thing. So the genome is small. And some diphtheria will toss out that gene and become avirulent. Well, that would have been a mess if they were culturing and then it didn't cause the disease and it wouldn't cause Koch's postulates. But fortunately for them, it did not. And so that they were able to, they had all three genes. So then they had that, they had the organism. When we talk about the toxin, and the toxin was isolated by Emil Wu and Andre Yersin. These are young guys in their 30s in Pasteur's lab. But Emil Wu, probably nobody remembers, but I remember from my last talk. Emil Wu, we know who Emil Wu is. We've met him before. Now, you'll recall, you heard my last talk, that Pasteur had attenuated anthrax by subculturing. And it lost its virulence. And the idea was, his idea, which was correct, was you could his idea was that you could then administer it, as Koch had the same idea, the avirulent strain. So, but again, Pasteur gave a demonstration of his attenuated bacteria, public demonstration, to show that it worked. He gave the real anthrax to one group of animals, and he gave the others attenuated anthrax. And of course, by the end of the day, all the animals that got the real anthrax, the real one, died. His attenuated strain did not. But in fact, what he did was, he used the method of a veterinary scientist from Paris, who had shown if you apply potassium dichromate to anthrax, it also injures the cell and attenuates it. So before he did his demo, he took his attenuated strain and applied potassium dichromate to it to make sure that it's attenuated. And he even put it in his notebooks that he did it, because then we got lab books, just like we did in lab. He asked that his book lab manuals never be published, but ultimately his family did publish it. He did do it, but unluckily for him, Emil Wu saw him doing it, and he wrote it down in his own notebooks that he saw the boss cheating and putting the potassium dichromate on the anthrax before the demo. So he went from being his minor young employee to being his partner, because he had the goods on Pasteur. So the great Louis Pasteur. Pasteur was like to bacteriology what Isaac Newton was to physics. And he had the goods on the boss for cheating.
So from an outsider's perspective, this question is, what difference does it make what actual method was used to extract a reduced function version of the bacteria? Why would he claim it? What difference did it make whether he made that claim or not? I mean, I see that it was fundamentally dishonest that he did make the claim, but why did he make the claim?
In French, it's like what? He said the glory is that you did it all.
But wouldn't it have been just as glorious to have another means of arriving at a reduced function?
Share that attenuation. Not if you have to share it. He didn't want to share it.
I see.
He wanted
the way he went about doing it.
Okay.
He wanted to say, I did it.
So he wanted to keep the method secret
Right.
for his own.
Not, oh, I took his model and I just gave it to the sheep. That's not good. These were very competitive people fighting their way. So there you go. So okay. So there you go with Pasteur knows it as well. But now I digress. So how are they going to do this? Well, what they used was the Pasteur Chamberlain filter, invented in Pasteur's lab by, he was Frenchman, Charles-Edouard Chamberlain, Chamberlain in English, Chamberlain in French. And I'm going to show you the filter, the diagmatic, and then I'll show you the filter. What he used was a porcelain filter. This is the case here, and the filter is here. This porcelain. Now, porcelain is very porous. The reason your teacup doesn't drip tea is because the glaze that's on the inside prevents the tea from getting to the porcelain. But it's very tiny, very, very tiny pores. And Pasteur needed this for his own laboratory. He needed bacteria-free water in the Seine, where they got their water from, mostly from the Seine, was filled with bacteria. Now, this is 1880. The significance of the bacteria in the water was not known. But they were there, and they would overgrow his culture. It doesn't matter. He needed sterile water. It's a pain in the neck every morning to stoke up the stove boil 20 liters of water and then let it cool and then get started. He wanted a more efficient method, and Chamberlain invented that. What he did was he made a slurry of water, percent, put it in here. The bacteria couldn't get through this porcelain membrane, but the water would come out. And that's the essence of how it works. So here's the sort of the manual mock-up first and the low production. Here you have, here's the main beaker here, and here's the porcelain filter here. Here's the porcelain filter. And they pour regular city water in here. And up here, we just have a syringe to create negative pressure. It doesn't matter whether it's positive pressure driven or negative pressure driven. We have suction here. So here's the bacteria in this water, in the city water here. Here's the city water. The city water is sucked in through the porcelain membrane, comes up here and into this bottle marked O pure, pure water. And that's how we got it. The actual device that they used for high volume was really the same principle. What he did was, there's a cutaway view there. Here's the porcelain filter here. Here's the tap here. Puts the tap water into here, screws on, drives it with positive pressure through the porcelain membrane and out here. And so you can just turn on the spigot and get back to your free water. And periodically they clean the filter, but very efficient. It is just the same device in this jacket. So that's the Chevron filter. Back to the wrench. Come on, back to the wrench. So how are we going to isolate bacterial toxin? Well, now this is a leap of thought here. We're going to talk about data and new models. We hypothesized that diphtheria bacteria produced a toxin which caused the disease. But he needed a method to separate the toxin from bacteria. And toxins have not been identified. This is diphtheria. So he used the Pasteur-Chamberlain filter. And what he did was, he took the bacteria, the toxin, put it into here, and then, oops, drop it. One more. No, I can't make it work. Huh? What happened was he would put the bacteria and the toxin here, and the toxin came out, it was driven out here. These small molecules. It collected the toxin, independent of bacteria. Giant leap. Because what he found was that the filtrate caused symptoms of bacteria, of diphtheria. The filtrate did. There were no bacteria there. So it wasn't caused by the bacteria, it was caused by the filtrate. And they actually had the word toxin at the time. So what he wrote in 1891 was, it is natural to conclude that microbes act through their chemical products, true poisons, specific to each of them, and which determine the symptoms of the disease of man and of animals. The infectious disease is therefore a poisoning. The source of the poison is the microbe settled in the tissues. It elaborates his toxin there at the expense of the living being it is going to kill. We got to know the cause of the disease before you get the cure. So they get closer and closer to the cause of the disease. Now he's got it. This is the biggie. So now diphtheria produces an exotoxin, just to give you the language. An endotoxin is bound to the cell wall, and it exerts its effect on bacterial cell death. It's an endotoxin. An exotoxin is actively secreted from the bacteria. So here you have the cell wall, it's just a living cell and bacteria, and it just pushes out the exotoxin. Whereas this bacteria, this other form of bacteria produces endotoxin, and it has to die before the endotoxin is released. So with Lou and Yersin, because the toxin was released by the diphtheria, they were able to filter it, keep the bacteria there. It would not have been an easy task had it been an endotoxin. It was luck. But they just happened to be that way. So there you go. And then we get to Von Behring and Shibasa Buro Kisata. Say that 10 times. And they published an article together. They were at the Koch's Institute on the development of diphtheria immunity and tetanus immunity in animals. Now, Von Behring was from Germany, and Shibasa Buro Kisata was kind of a postdoc. He came to the Koch Institute from Japan to learn science. France was that time the center of the world. It still is. It was the center of the world of science, and so he come from Japan to bring back to Japan what you learned. So he was basically a postdoc. Now this is what he did. He did this. But Behring had nothing to do with this. Kinsano worked with tetanus. But tetanus was thought to be impossible to culture because without the tiniest bit of oxygen, it wouldn't survive. It's an obligate animal. And what he did was he worked up a Rube Goldberg machine that pumped hydrogen into it with a little positive pressure. So the oxygen could never, might have some leaks, the oxygen could never get in. And so he was the first person to successfully culture tetanus. And tetanus produced a hot toxin. And he, using the Chevrolet filter, he isolated the tetanus toxin. And now he takes, develops an antitoxin. Now this is like a giant thing to do. He takes the usual suspects, the guinea pig and rabbits, and he injected them with sublethal doses of tetanus toxin. They know nothing about immunology. The animals developed, they developed some antibodies, became immune. We know that. He didn't know that. He just noticed that they could tolerate it. And he gave them escalating doses. And they kept tolerating it. And then he took the serum from one animal, put it into another animal, and the other animal was resistant. So he had discovered something. He had taken antibodies and transferred the antibodies from one animal to another. A giant step. And here's what he did. He took tetanus toxin and he gave it to a rabbit in escalating doses. Of course, the rabbit creates more and more antibodies so it can withstand the toxin, and it gets into what we call now a hyperimmune state. And then he took the serum from the rabbit to another rabbit, and lo and behold, he gave it a toxic dose of tetanus. The reaction. So now he had transferred immunity from one animal to another. He had a hyperimmune state, transferred immunity. This was his research. Okay. So Von Behring, also in the lab, he was an established scientist, he joined forces with Kinsano, and they applied Kinsano's technique to isolate the diphtheria toxin in most rabbits and guinea pigs. So they did the same thing that he had done to tetanus. They just used diphtheria, which had been now they know how to culture it, and they could do that. And this is some honorary thing that I swiped from somewhere. So you have diphtheria toxin, which is a healthy rabbit. They use the word vaccinated. It isn't vaccinated, it's immunized. Diphtheria resistant. They take the serum from the blood here, give it to a healthy rabbit. The rabbit lives. They give virulent diphtheria to a healthy rabbit who's not immunized. The rabbit dies. Same as with tetanus. Now, rabbits are too small. They don't give enough serum for administration to humans. But horses are the same. So they could give serial injection of toxin to horses and give large volumes to be harvested for human administration. And Von Behring gave it to humans. His results were kind of mixed. He didn't know the dose. He didn't really have it down. He was pretty sure it would work. He didn't have it down. So back we go to our old friend Emil. And Emil Roux, he does the same thing in making more serum. And he takes varying doses, and before he does the study, he just empirically looks, how much did it take? How much did it take to cure the disease? So he figured out the dose, just empirically, there's no measure. And once he figured out the dose, he did a study at L'Hôpital Necker. Necker is the lady who funded this hospital, children for a hospital for sick children. And he took just 300 kids, and he had the dose right, and he reduced the mortality from 50% to 10%. Uncomplicated by other bacteria. That was an enormous effect with theory. And I love this as a physician. Look at this. The Petit Journal is Le Croup guéri. The croup is conquered by Dr. Roux. Now look at this. Here's Dr. Roux in his suit there, administering. They just gave it subcutaneously. That's a difference. To the kid. Look at the mother. She's not looking at her child. She's looking at Dr. Roux. Is that great or what? What could be better than that? And the distraught father, the distraught maid, but you know the kid's going to be sick of it. The title tells you that it's conquered by the group. It's conquered by Dr. Roux. So Roux gets the good publicity. So how much did they really know? Nothing like you think. Now, Kendall Smith wrote an article on Louis Pasteur, questioned the father of immunology. Pasteur was entirely mistaken as to how immunity works. This is like Isaac Newton not understanding physics. In that he reasoned, as a good microbiologist would, that appropriately attenuated microbes would deplete the host of vital nutrients, like a culture plant, absolutely required for their viability and growth, not an active response on the part of the host, that we were just depleted of some essential nutrient. Roux and Chamberlain, however, wrote, no, they could write because he had the goods on Pasteur. Monsieur Pasteur, that's their boss, thought that in the case of foul cholera, the non-recurrence was due to the disappearance of some substances consumed by the microbes. It is not necessary for the cells of the pathogenic organism to live among the cells of the animal to confer immunity to it. So it wasn't a consumption of vital nutrients. Boss. So here's Pasteur surrendering to Roux. This is the tone. Speak if you want of poisoning. Make this hypothesis. I accept. I do not know the mechanism of death from any disease more than you or anyone else does, no more than we know the mechanism of life. Speak of poison if you wish, but you will be forced to add that if a poison causes death, it is the microbe that generates the poison. I'm still here. I'm still here. So we got his boss. Now I should mention that you just heard it, serum sickness from horses. Listen, the antibodies you get from horse serum, they hang around a long time. And we get antibodies to them. They precipitate joints, skin, blood vessels, kidneys, heart. And it occurs a couple of weeks after administration of the horse serum, build up your own antibodies against the horse serum. But people are kind of sick, but mostly they just get better. It's good
Has autism.
it does do that. So Koch married Hedwig Freiberg. She was a 20-year-old medical student. He was 55. And then to be done Emil von Behring married Elsie Spinola, age 20. You look at the two of them, they kind of look like they were separated at birth. It's interesting. We thought to bring it up to a modern day to see how it comes out in modern.
Yeah.
So
Wait a minute.
Yeah, that's what I was thinking. It's the Bill Belichick effect.
as we say in French, plus ça change, plus c'est la même chose, which means the more things change, the more they are the same. But listen to this. 1 week after the von Behring-Kinsotto paper, which was very important, von Behring published a single-author paper, Investigation of the Mechanism of Diphtheria and Immunity in Animals, at which point he writes in there, the possibility of cures, meaning by me, the possibility of cures for even highly acute disease can no longer be ignored. Kinsotto was unaware of that work. They got published in 7. And you gotta know that. Do the science, you had to write it up, you had to public peer review, rewrite it, get it to the printer, publish it. Came out 1 week after his paper with Kitasato. Kitasato did not know anything. So let's go a little deeper. Emma von Behring's Nobel Prize speech. No mention of Kitasato. He gave a dinner speech after at the end of the Nobel Prize ceremony. No mention of Kitasato. As far as he was concerned, this was the team.
You think this was systemic racism
Yeah.
thing? You think it was a systemic racism thing?
Well, let's just see.
I'm not jumping to conclusions, just.
Here's what it is. Follow the money. Spoils of war. Von Behring took the whole 8 million Swedish krona all for himself. Not only did that, he then worked out, now Hearst is still around, it's a drug company. And he worked out constructing a subsidiary called Behring's work, von Behring. He received 50% of the profits from the sale of diphtheria antitoxin. Kitasato got exactly 0. So, and Behring's work now is a big company, it makes lots. Kitasato
Oh, go ahead.
versus von Behring. There were disagreements, I'll say it lightly, over who deserved primary credit for the antitoxin work, leading to a strained relationship. Kitasato versus Koch. Koch favored German scientists for appointment.
Yeah.
Kitasato had done this enormous contribution, was a postdoc, and he wasn't going to get appointed. Because he thought he was. So Kitasato packs his bags and returns to Japan, and he made no comment about his omission in von Behring's Nobel Prize speech as for not sharing the 1901 Nobel Prize, for which he was nominated.
He was nominated.
He was nominated.
Interesting.
Yeah. Von Behring versus Koch. Koch was initially invented von Behring, but von Behring's work diverged because Koch was failing to cure tuberculosis with tuberculin, driver TB, while von Behring was successfully curing patients with diphtheria. And von Behring's success began to overshadow Koch's failure. And neither man publicly acknowledged the other's work, and Koch did not support von Behring's candidacy for the Nobel Prize. These are tough guys. Now, I'll just finish up with the rest of the story. Toxin to toxol. A toxin is a poisonous substance produced by a living organism. Toxoid is a toxin treated to destroy its toxicity while maintaining its immunogenicity. Oid, of course, means likeness. And Gaston Ramon in Koch's lab, what he did in 1922, he combined serum and antitoxin from other people. He got a precipitate called a flocculant, really tough to boil. And so the antigens and antibodies get together and they precipitate the flocculant too. That's the flocculants. And that tells you how much you have. So if you put one in excess of the other, then that major excess, then the amount of flocculants will be dependent on the one that's not to excess, because all of it will be bound and that will be it. So you can figure out what you have. The concentration of antitoxin correlated with the amount of precipitate, and the dose of serum antitoxin would be quantified. Now, everybody knew that if you took formalin is formaldehyde and water, 50-50. That everybody knew that it made all proteins, everything. So he did it to the diphtheria toxin. What he found, here's a discovery, that the toxin still precipitated with more serum. So he reasoned it must look like to what this magic stuff in horse serum is. Must look the same. And the inert toxin could be used to generate tolerance to the toxin. So now you could give a non-toxic product. You didn't have to do all these escalating doses. You just start giving the toxin. And you could determine the effective dose of toxin by the amount of precipitate that was produced. And this led to direct immunization of humans. The suitable dose of toxin It's also called antitoxin. Now look at how fast he did this. 1922, he noted the toxin with hormone losses toxicity, but maintained its ability to induce population. 23, he discovered that the toxin maintained its ability to produce immunity to diphtheria. And in 24, he began inoculating humans. Boom. Between 1930 and 1953, Ramon was nominated 155 times for the Nobel Prize, and he never won. Well, feet of clay. You do an investigation, you find out this. Ramon willingly produced tetanus, he was French, tetanus and diphtheria vaccine for German soldiers during World War II. He was investigated but not officially determined to be a collaborator with the Germans. There's a lot of conjecture that he may have limited his appeal to the Nobel Committee. And we've all had DPT or Tdap, which is tetanus, diphtheria, and pertussis, or TD, you can get that as well. But what we've done is we've generated herd immunity. It's past man to man, and it's just too much herd immunity. So look at the comparison, the impact of what these people did, especially here at home. In the 1920s, 100,000, 200,000 cases a year. 1996 to 2018, 14 cases of diphtheria, 1 death. 1 death.
Is that US or worldwide?
United States. And there's a diphtheria horse serum. You only get a CDC because there's no call for it. But you can still get the same horse serum. And I just want to finish up with local guy makes good. Drew Weissman, Lexington High School, 1977. He won the Nobel Prize. RNA, if you inject it, you put it into a cell, it's rapidly metabolized. This is wrong. So he substituted pseudouridine for uridine, and the enzyme for degrading RNA doesn't work. When the RNA is pseudouridine, even the RNA works. And he developed nanoparticles in the nervous system. Won the Nobel Prize. And that is it. Good. You know all about this theory. Information, $2.20, you can take the subway all the way from Alewife to Boston.
I get it cheaper.
I get $1.10 because I got
You got the senior discount.
I do.
Yes.
What did your dad say at the end of this presentation? Would
So
he,
we're back on. Yeah.
would he recognize the positive?
We're just back on, Steve, right? No, I'm just.
So when the Nobel Prizes started up, you said it was 1901, is that right?
It was awarded in 1901. It was established in 1895. You put the money in.
First one was 1901.
What was the initial, whatever, taxonomy of awards? Did they have
They-
biology and
Yes.
chemistry
They
and
had
physics? and-
everything except economics. They had medicine and physiology, chemistry,
Or medicine.
medicine or physiology,
That's what it's called.
physics, chemistry, peace
Literature.
prize, literature,
Right.
peace, and then
No, yeah, please.
peace, and economics was added in 1960.
Okay, so economics is the only real significant add-on. Okay.
So actually, there were 5 given in 1901.
Yeah.
So first in physics was Röntgen, first one physiology or medicine.
Right.
Yeah.
It was barbaric.
The
We didn't think so.
diphtheria one.
We didn't think so, but
Yeah, so initially there weren't 5 prizes that were granted because the committees couldn't agree on anyone except Röntgen. Röntgen was viewed as, a angel from another planet bringing X-rays into the world. And
yeah,
then they
of course.
started realizing that you didn't have to be quite that. And we ended up with starting very early, actually, getting a woman involved. But that was a challenge, getting Marie Skłodowska-Curie involved. If it wasn't for the intervention of Henri Becquerel, only Pierre would have gotten the prize. And he said, no, she did as much as Pierre did, and they go, well, that's impossible because she's a woman. And then after Pierre got knocked off by getting hit by a wagon, they gave her a second prize all on her own. And then the family ended up with five Nobel Prizes in the family, which is unprecedented.
We were in the ivory tower. There's a lot of money to be had up front and then downstream. So commercialized product. I don't know if Rankin commercializes product.
But it's amazing how everything was concentrated in the hands of Germany and France until basically the British finally broke through about 7 or 8 years into the sequence of prizes, and that was strange because there was a lot of very good work going on.
Only
And,
only
in
to
England.
thank the Nazis for driving out all the Jewish scientists who came to America.
When was
Not
it?
that we never drive out the scientists. We never do that.
Do you happen to know when the first US Nobel Prize was awarded?
I wouldn't know.
Anybody have any idea?
I'm guessing it was Michelson and Morley.
If not, and if not them, it was damn, I just dropped the name. Millikan.
But we could certainly look it up.
Oh, yeah,
It could have been
otherwise it was
Millikan. Millikan was a little bit later, I think.
right in the
But
20s.
it's probably somebody more obscure, but
In 1900,
anything comes up here, first American.
You know, infrastructure. Yeah, man. We had engineering and building machines.
Yeah,
We did the
industry.
World's Fair at that time.
Huh. Okay, well, the first American Nobel laureate was Theodore Roosevelt for the
Yep.
Peace Prize in 1906, for ending the Russo-Japanese War. Michelson wins in 1907.